Walden Butterman.
Good morning to all. Good morning, everyone. Rayat Shikshan Samsthas, Art, Science and Commerce College, Mokhada, and IQSA are jointly organized this today's international online conference on biodiversity conservation and the role of molecular phylogenetics in the plant systematics 2022. So myself, SY Tide, welcomes you all for this international conference. As we all know, our college is situated in the hilly tribal. As we all know, our college is situated in the hilly tribal region of the Western Ghats in the Maharashtra. Our main aim for organizing this conference is to make awareness in the tribal peoples about the biodiversity and its conservation. The tribal peoples are very aesthetics about nature as they save the plants and animals in aesthetic ways. They serve nature as a god. That's why we are the most biodiversity found in the areas like Mokhada. So it will give it will be the great to inform you that the conference in this conference more than 350 people are registered all over the world. More than 60 participants are going to present their work about the biodiversity conservation and the molecular phylogenetics role in the systematics. Participants from the countries like Nigeria, Sweden, South Korea, and also joined in this conference to present their work. From India, people from Karnataka, Kerala, Gujarat, MP, UP, and all over the India are joined us here. So heartily welcomes you all again. And now I will request Dr. A.N. Sandore sir for their inaugural speech. Over to Mr. A.N. Sandore.
Good morning to all. Rayat Shikshan Samsthas, Department of Botany, Art, Science and Commerce College, Mokhada is organizing online international conference on biodiversity conservation and role of molecular phylogenetics in plant systematic 2022. Mokhada is unique place situated in the Western Ghat and majority peoples belongs to tribal community. Biodiversity hotspot is a region that is a prime location for the existence of rich biodiversity, but also faces the threat of destruction. It is a place that needs our immediate and constant attention to survive and thrive in the future as well. All over the world, 36 areas qualified as a hotspot. Their whole habitat represent just 2.5% of Earth land surface, but they care more than half of the world plant species as endemic, and nearly 43% of bird, mammal, reptile, and amphibian species are as endemic. India has always been on list of richest country in the world for its biodiversity. There are two biodiversity hotspots are present in India, which includes the Western Ghat and the Himalayas. Western Ghat is one of the hottest hotspots of biodiversity in the world, and there are more than 6,000 vascular plants here, which belongs to more than 2,500 genus, and out of these 3,000 plant species are endemic. The Himalayan comprise more than 100 mountains beyond 7,200 meters. There are about 10,000 species of plant in which Himalayas, a third of which are endemic and cannot be located anywhere else in the world. Western Ghat and Himalayas are under huge pressure, and number of species are rare, endangered, and threatened category. In recent years, increasing loss of biodiversity has created serious threat to the survival of mankind. The main causes of biodiversity loss are habitat destruction, pollution, population, invasive weed, overexploitation, and climate change. Destruction of the habitat is the major threat of biodiversity. Therefore, it has become necessary to conserve endemic and threatened plants of hotspot region and in India, especially the Western Ghat and Himalayan region. The main goal of this conference is to develop awareness among students, researchers, and teachers about biodiversity and conservation measures for rare plants. Thank you.
Thank you so much, sir. Now I will request to Professor S.S. Sandanshu, sir, the convener and the head of Department of Botany, to address us about the welcome speech. Over to Professor S.S. Sandanshu, sir.
So I would like to welcome all eminent personalities participated in this conference on behalf of our department as well as the college. First of all, I would like to welcome Dr. S.R. Yadav, INSA Emeritus Professor from Shivaji University, Kolhapur, Maharashtra. Then Dr. Asif S. Tamboli, Department of Biology, South Korea. And our Honorable Principal, Dr. L.D. Bhor, Principal of our college. And I would like to also welcome all the participants of this conference on behalf of our department and as well as the college. Just you know that the plant ensure a resource for new food crop and medicine. Plant life balances ecosystem, protect watershed, mitigate erosion, moderate climate, and provide shelter for many animal species. Threat to the plant biodiversity include the increasing human population, pollution, deforestation, species extinction. The plant extinction is progressing at an alarming rate. This in turn affect other species, which also become extinct because they depend on the delicate ecological balance. Effort to preserve plant diversity currently include heirloom seed collection and barcoding DNA analysis. The conservation of biodiversity in natural heritage site, including sacred groves, protected areas, and other biodiversity hotspot, is crucial for maintaining resilience of ecosystem. The lagoons of Western Ghat and Konkan are a very unique ecosystem harboring wide diversity of flora and fauna, which needs to be conserved on top priority. We know that on the other hand, the traditional classification of plant into respective classes, order, families, genera, and species has until recently based on morphologic, cytological, biochemical, and ecological trait. The technique in molecular biology have provided many tools for investigation of phylogenetic relationship among the plant. I would like to stop here or conclude here the welcome speech. Thank you. Thank you. Once again, welcome to all participants of this conference.
Thank you so much, sir. Now I would request our principal, Dr. L.D. Bhor, sir, coordinator and the principal of ASC College Mokhada, for give their presidential speech.
morning. Welcome all of you. Today, Section Sanskar, Arts, Science and Commerce College, Palagar, Department of Botany has organized one-day online international conference on biodiversity conservation and role of molecular biologics in plant systematics 2022. First of all, I would like to welcome Honorable Resource Person, Professor Dr. S.R. Yadav, from Department of Shivaji University, Kolhapur, Dr. Asim Tamboli from Biology, School of Life Science, South Korea, with, who have present here from the UG city. I would like to welcome our partners, Honorable Sharad Chandrajit Pawar Sahib, President, Ayat Shikshan Sanstha Satara, Honorable Dr. Sahib, Chairman, Ayat Shikshan Sanstha Satara, Shinde Sahib, Vice Chairman, Ayat Shikshan Sanstha Satara, Honorable Absenti Thakur Sahib, Member of Managing Council, Ayat Shikshan Sanstha Satara, Chairman of College Development, Shivankar Sahib, Secretary, Ayat Shikshan Sanstha, all participants. I also welcome organizer and lab coordinator, Dr. Sir, Professor Selvashi Sir, and Professor Aide Sir, has organized such important conference on current issues in the tribal area like Mokhada in Maharashtra. Dear friends, biodiversity means diversity among living organisms, which includes bacteria, fungi, plants, and animals. All these organisms are essential for sustainable development. But nowadays, due to the nature of human being, many organisms exist on Earth and some are on the way of extinction. So we need to conserve them. In this conference, we will discuss about another issue, that is conservation of biodiversity. Plants are very important for the maintenance of ecological balance because they are producers. When we concern with the human being, national development, we all are dependent on plants for food, shelter, and service as well as medicine. By keeping this need in mind, we are organized here for this conference. Finally, I would like to wish all the best of this online. Thank you.
Thank you, sir. Thank you so much. Now, we are going to the next session, Keynote Address. Now, I will request Dr. N.V. Malpure, sir, SSGM College, Kopargaon, to introduce about today's keynote address, a person, Professor Dr. S.R. Yadav, sir.
Good morning. Can you hear me?
Yes, sir.
Yes, sir.
Yeah. Good morning. Dear all, it gives me immense pleasure and I have the honor to welcome and introduce today's speaker for this session. This keynote address will be delivered by Honorable Dr. S.R. Yadav, sir. Actually, he don't need any introduction, but as a protocol, I have to do it. Honorable Yadav, sir, served as professor in Department of Botany in Shivaji University from 2005 to 2016. He worked as INSA senior scientist in Shivaji University. He has several awards and honors to his credit. To name some of them, like E.K. Janki Ammal Award, which is given by Ministry of Environment and Forest, New Delhi. Late Professor V.V. Sivarajan Gold Medal. Then Dr. Ek Bote Prize, which is given by Agarkar Research Institute. Then Father Santapoy Medal, and there are many more awards, list goes on. He is fellow of Indian Association for Angiosperm Taxonomy. He is fellow of INSA, Indian National Science Academy. He is fellow of Maharashtra Academy of Sciences. He is fellow of National Academy of Sciences. He has guided more than 11 students for MPhil and 26 students for PhD. He has published over 270 research papers in national and international journals. He has completed over 20 research projects, and the main project which I would like to mention here is he has successfully established lead botanical garden at Shivaji University, Kolhapur. He is, and as you know that he is well-known taxonomist of this country, and he is known by the world taxonomist. His students has honored him with describing many new species on his name. To conclude here, now I would like to invite and welcome Professor and Honorable Dr. S.R. Yadav sir for this to deliver his keynote address. I welcome him to deliver his keynote address. Thank you.
Thank you so much, sir.
Kindly allow me to share my screen.
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir. Now you are host, sir. You can share screen, no problem.
Is it visible now? Hello?
Yes, sir. Yes.
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay.
Now I request to Professor Dr. S.R. Yadav, please give today's conference keynote address.
First of all, I would like to congratulate this college which serves rural India, student from tribal community, and that is Mokhada, which is a place usually people think that it is a very remote place and nobody likes to go there. But all those teachers who are working in Mokhada College, I appreciate their efforts and their sincerity to work for students from rural India. Similarly, I appreciate Dr. Bore. Hello. Kindly mute yourself. Those who are talking, please kindly. Hello.
Arun sir, you please unmute all.
Dr. Anjali Thete, ma'am, please mute yourself.
All participants are requested, please unmute yourself.
Mute yourself.
Dr. Anjali Thete, ma'am, please mute yourself.
Okay. So I think otherwise we will not be able to listen. So your
please mute yourself.
Dr. Bore, who is leading the college, principal of the college, and I appreciate his efforts and commitment to students from tribal community. Similarly, our Sandan Shiv, who told about conference. This conference will be definitely of some use to the students, and that is the purpose of this conference. So another thing that Dr. Malpure introduced me, and definitely he is my student, so definitely he has a lot of love, respect for me, and he introduced. I am thankful to him. So today I will be talking on a topic of concern to everyone, is taxonomy and its role in documentation, bioprospecting, and conservation of biodiversity. First of all, what is biodiversity? Almost all of us we know, since this word was coined in 1985, most of us we are aware of biodiversity, its importance in our life, and need for conservation. So that is known to all of us. Now we know when we talk about plants, especially the trees which stand outside your college campus, one single tree does work of about 3 lakhs rupees per day, per year. So ecological services provided by any tree, medium-sized tree, goes up to 2 to 3 lakhs per year. And that way, in addition to fruits, other benefits, direct benefits, it provides ecological services. And really speaking, our survival, our progress depends on biodiversity. Now, you know, one thing which I have studied as a student of botany, that is, plants are most essential for the existence of animals, including mankind. Without plants, we cannot exist. Of course, somebody may say that, sir, sunlight is important, sun is important, soil is important, water is important. Of course, they are important. But in between these two, animals and other people, I mean, other things, plants stand in between.
Sorry, sir. Mr. Govind Pendekar, please mute yourself.
Now, second thing which I have understood, that is the true matrix of human life, is the green world covering Mother Earth. Since morning, when we take tea, we start with our plant, and till we go to bed, we sleep on the bed of cotton. So 24 hours, plants are with you, and that way plant has their own significance. Similarly, another very important thing which I have understood is a fascinating physical, emotional, and spiritual relationships that exist between plants and man. You know, it is not just direct benefits which we take, but another part of our life is our physical, emotional, and spiritual relationship is very deep with nature, with plants, and that way they are of great significance in our life. You know, this is a sentence which I have written. You know, without green plants, we would neither breathe nor eat. This is the truth. This is Malabar frog. Whether Malabar frog will survive without food, without air. So therefore, plants are of great for our survival. You know, whether insects are there, butterflies are there, snails are there, frogs are there, birds, mammals, reptiles, whatever, you know, and human beings, our existence depends on plants. I hope you understand what I mean to say. The very existence of human being depends on plants. What progress we do, what technology we develop, that is a secondary. Primary, for our own survival, these plants are most important, and only when you survive, you can enjoy all technology, all modern developments. And therefore, this is of basic importance in our life. Now, this is the Mother Earth, which is the only planet which has plant diversity or living world, biological world or organic world. And this is the only planet we have life on it. And you know, this slide I usually show, reason is our origin. When we talk about our origin, this hominoidea, this branch, this clade originated sometime 20 million years ago. And the first branch, it branched and it gave rise to gibbons. Then another branch it gave rise to, from second branch is orangutans. Then third, about 8 million years ago, gorillas got separated from this clade. And about 5 million years ago, chimpanzees got separated from that clade. And finally, about 2 lakhs years before, or 0.2 million years before, Homo sapiens or genus Homo was originated. So this is our history. So from Gibbons up to Homo sapiens, we have, evolution has taken place, and our history is only 2.2 million years. Now see, we originated in this Rift Valley. We were there in Rift Valley for about 1.5 lakhs years. And after that, we started migrating to other parts of the world. So chimpanzees are our close relatives. All of you know molecular biology has helped us to know what are the similarities between chimpanzees and human beings. About 98% of DNA is exactly similar to chimpanzees. So in other words, 98% we are chimpanzees and 2% we are Homo sapiens or human beings. And we are the latecomers on this planet. This species, Homo sapiens, is the very latecomer on this planet. So this is what I wanted to tell you. After from this, origin in Rift Valley, after 1.5 million years before, we started migrating to various parts of the world. So first we reached Australia, then to this Eurasia, then North America, and then South America. In South America, we have reached only about 10 to 12,000 years before, and Australia 50,000 years before. So human history on this planet is only of 50,000 years. We occupied this planet, Mother Earth, in last 50,000 years, and with increasing population of Homo sapiens, we started agriculture. So about 10,000 years before, we started agriculture, and all of you know, we human beings are every day, every second, we are claiming wild land, terrestrial land for agriculture. And most of the land is now converted into agriculture. And this agriculture from whom? This agriculture is only for human beings and some of the animals, domesticated animals. So where other organisms will go? So that is the question. And agriculture is the major cause for loss of biodiversity. Every day in Western Ghats, you are near to Western Ghats, you know, the forest areas are converted into agricultural lands, and that way the wildlife is threatened because of our activities. At the same time, we are very clever and brilliant. If we destruct, we can construct also. You can see this diversity in fruits, vegetables, all that diversity we have created is also amazing diversity, but we have to care for other biodiversity also. Now, if we see what is the magnitude of biodiversity, how many species we know. So we know about 1.75 million species, or other words, 1,750,000 species we know. 1,75,000, 17.5 lakhs species we know. And the fair estimate of total biodiversity is 13.65 million. That means 136.5 lakhs of species are supposed to occur on this planet. Now, out of this 136 lakh species, we know only 17 lakhs. So our knowledge of biodiversity is very, very, very meager. So out of those total species, we have studied only this sector, about 17.5 lakhs of species. Remaining biodiversity is yet to be studied. So we know only 11% biodiversity. Remaining 89% biodiversity is yet to be studied, yet to be understood, and that is our knowledge of biodiversity. Now, you know, when we talk about biodiversity, and the conference is on biodiversity, its conservation, first problem which we are facing today is biodiversity loss. All over the world, we are losing biodiversity at a great rate. Okay, great rate we are losing biodiversity. all over the world, there are crises for biodiversity. Food crisis, water crisis, and people are not getting food to even eat. And that way, biodiversity crisis. And we have entered into 6th mass extinction phase. Mass extinction, species are lost in masses, in large number, and that is a mass extinction. So these are major issues with reference to biodiversity which world is facing. Yesterday's news you must have seen in India, in Nagpur region, we have a locality, Brahmapuri, which has highest temperature in the world recorded. And that way, things are happening due to biodiversity loss. So importance of biodiversity. Biodiversity is the very basis of human survival. Our survival depends on plants, and it is a underlying cornerstone of progress. Progress does not mean roads, or this aerodrome, or any technological development. Real progress of human beings, real pleasure of human beings, peace of human beings lies in biodiversity. So that is the cornerstone. Already, there are now at present 36 hotspots of biodiversity have been declared, and in India we have 2 or 3 biodiversity hotspots, and we are in Western Ghats, and that way we are concerned deeply with that. There are about 2.5 lakhs of flowering plant species estimated. Only 30,000 species are edible. 7,000 have been used for food, of which 120 are cultivated today, and some 9 provide more than 75% of human food. 9 plant species, and out of that, 3 species like wheat, maize, and rice provides more than 50% of human food. So we are only using 3 plant species out of 2.5 lakh species as a food, as a major food. Similarly, grasses, legumes are of great significance for human survival. So I will say something about taxonomy. Taxonomy is a branch of biology which has a function of documenting biodiversity. So what we do, classical taxonomists, that is they collect plants, they identify, that means we document, then we classify and give some name, and that name is of great importance, and taxonomy is a central, has a central position in biological sciences. Okay, and it has different aspects. Today I will be talking about classical taxonomy, which has a great significance today for every one of us. At the same time, we study processes of evolution. Then today, Dr. Asif is going to talk on phylogeny with reference to molecular data, and that is advanced fields of systematics or taxonomy. Now, I consider systematics or taxonomy as a wheel of biology. Without this wheel, biologists cannot do any progress, and that's why taxonomy is considered as a mother branch of biology. It is also considered as the axis of biological sciences around which biology rotates. And taxonomy is a foundation for biological sciences. It forms foundation for progress of biology, and therefore taxonomy has a great significance. Now you see this is a karst plateau, and many people thought that taxonomy is the victim of its own success. Quite simply, scientists, so-called modern scientists, they say, and similarly public also take it for granted and think that everything in taxonomy has been done. Is it a truth? Everything has been done in taxonomy. Is it a truth? When I told you that 90% of biodiversity is yet to be discovered, documented, named, and classified, how you say that everything has been done? But unfortunately, some people who are specialized in some branch of biology, and they feel that they are the only people, they know science, and underestimate importance of classical works like taxonomy. Then, some scientists think that taxonomy has no value and it is unnecessary complication. So many of us, what is there in taxonomy? It is unnecessary complication. Some people even utter these words, these sentences, and scientists do not fully understand that taxonomy is a continuous, rigorous, and painstaking process of incorporating new information. So that way, it has a great significance. And, taxonomy is a lays solid foundation for conservation. Only because we know plants through taxonomy, we are able to take steps for conservation and then CBD, which is a Convention on Biological Diversity. If you want to implement, taxonomy provides basis for implementation of Convention on Biological Diversity. Similarly, global taxonomic initiatives all over the world, we have to document biodiversity, and the world has taken steps, and those are known as global taxonomic initiatives to promote taxonomy in the world. Similarly, global strategies for plant conservation, strategies we have to develop because we don't have that much fund to conserve. So those which are most important, critically endangered species, they have to be conserved, and that is known as global strategies for plant conservation. Different strategies have been implemented, the basis of taxonomy. Systematic Agenda 2000 and 2020, these are important aspects, and final goal is building on hidden opportunity to achieve the Millennium Goals, poverty reduction through conservation and sustainable use of biodiversity. So this is the way it goes. Then taxonomy lays a solid foundation for red list of threatened plants. That is a taxonomy only makes. Then taxonomy is a basic to understanding of biodiversity. Taxonomy and without taxonomy, documenting biodiversity is difficult. This is the formidable challenge for taxonomists of the century. And our knowledge of biodiversity is only 10%. Remaining 90% diversity is yet to be studied. Okay. Then the critical importance of taxonomy for conservation and sustainable use of plant diversity has been understood by many people. All conventions, initiatives, and strategies stress the importance of taxonomy. So you see, taxonomy is the lonely voice speaking on behalf of interest in biodiversity. In the sense, this is a sentence by Haywood in 1967. Since 1967, we are talking, taxonomists are talking for the conservation of biodiversity. Now only we talk a lot after about 60, 70 years, but this has been said long back, and that was a lonely voice that time. So the role of taxonomy in implementation of Convention on Biological Diversity is important. Writing floras, floras, monographs, revisions, these are of crucial importance to take steps in policy making, in legislation, and in conservation of biodiversity. So these are the words. The nomenclature is another very important, and it is a democratic process. It is not a haphazard process. One should understand, and you should not get annoyed with changing names of plants, because that is a democratic process. Bringing taxonomy to users is our duty. And taxonomy and conservation of island floras, Karst Plateau is something like island, and these floras have to be conserved. Role of taxonomist in conservation of critically endangered vascular plant. Who knows? Only biologists know what are the critically endangered plants and animals, and we have to take steps for their conservation. So similarly, there are many species in wild which have become very, very rare. We have to reintroduce them and restore the populations of those species. That there also taxonomy plays important role. Taxonomy and human progress are closely related, and molecular systematic is another way for measuring and monitoring diversity. That is the most modern branch in biology, which is playing important role. In legislation also, when we make rules for utilization of biodiversity, conservation of biodiversity, taxonomy plays an important role. And, gardening of earth, the contribution of botanical gardens to plant conservation and habitat restoration. I have request to our principal, Dr. Bohr, that in his college, if he can take steps that in his college campus, if he can conserve some of the rare plant indigenous trees, that will be wonderful contribution by educational institute for the cause of conservation. So botanical gardens have to be developed by each college, each university, and that way we can conserve some of the plants in our botanical gardens. Taxonomy forms a framework for botanical gardens, and we need taxonomies. Similarly, wild seed banks is a new concept, and people are conserving plant diversity in the form of seeds, which is easy. They need less space, and we can conserve them for many years, and that is a wild seed banks which people are developing. So similarly, there is a need for support to taxonomy, infrastructure, and funding. Now, 21st century already our Professor Sandan Shiv and our Dr. Chandore has told what are the problems of 21st century. So one by one, age of mass extinction, then anthropogenic activities and pressure on wild resources. Then we have explosion of human population. Then we have food security problem. Then we have biodiversity crisis, water crisis, energy crisis, and invasive weeds, which have come to the which are creating a lot of problems to our wildlife, and then pollution, industrialization, climate change, increasing CO2, and this global warming, and many more problems which I can mention. These are the problems for loss of biodiversity. And all these problems are man-made problems. And if you want to have solutions for these problems, solutions lies in especially biodiversity and particularly in plant diversity. Okay, if you want to bring down carbon dioxide of atmosphere, we have to plant more trees. There is no other way, other technology by which we can bring carbon dioxide lower down carbon dioxide in atmosphere. So threatened species, endemic species, hotspots, sustainable development, these are buzzwords, but how many of us we know what are the threatened species, what are endemic species, where they grow, how to conserve, all that knowledge we don't have. And conservation of biodiversity is a challenge for 21st century. Now, role of biologists, taxonomists, systematists, botanists, zoologists, ecologists, what is their role? All of us, we know biodiversity, we know where it exists, we know what are the reproduction in plants and animals, how they reproduce, then what is the field status of species, what are the growth requirements of organisms, and then what are the conservation priorities. Then we know ecology and growth requirements of species, and that way, we know methods of propagation, captive breeding. We will also make biodiversity registers, advise in conservation, and finally, I would say the major role in framing of legislation with regard to biodiversity is done on the basis of knowledge of biodiversity, which is with biologists, who has a major role to play in sustainable development. To all these questions, there is a last answer that biologists, especially taxonomists, are of prime importance in 21st century to provide solutions to most of the acute problems of the world. So 21st century, will be dominated by biological sciences, and we have challenge of biodiversity documentation, conservation, bioprospecting, sustainable use. All these are the challenges for biologists of 21st century. Now, every species is one thread if you take, and if this thread gets cut or the species become extinct, in that ecosystem web, there will be big holes, ozone hole, all of same way, this entire ecosystem, this web will collapse if we lose our species. And it is said that by 21st century end, we will be losing 50% of our total biodiversity, which is a great danger, and even it threatens very survival of human beings. Okay, so each species is one thread of an ecological fabrics, and that has to be conserved. In biodiversity documentation, conservation, and sustainable use, we need taxonomists, we need ecologists, we need reproductive biologists, we need microbiologists, we need sociologists, we need politicians, we need leaders, we need policymakers, and we need foresters, and similarly, we need international cooperation. So no one alone can do conservation in meaningful. It is a multi-dimensional problem, and we need many people to get involved in conservation of biodiversity. Then only we can do, and especially society, our local people are of great significance or importance in conservation of biodiversity. Okay, so biology, paramount role of biologists is there in documentation, conservation, and sustainable use of biodiversity. Taxonomy is a cornerstone and in sustainable utilization of resources. Now, these are the questions which I have. These are the things which we are supposed to do: documentation, conservation, sustainable use of bioresources, implementation of CBD, global strategies. These we have to. Now, you see this. In addition to these things, conservation, really, the plants are wonderful creations of nature. And every plant has beautiful flowers. They are a vital component of world's biodiversity. You see the diversity. They are most essential resource of the planet. And, nearly 34,000 species are declared to be threatened by IUCN. And that way, not by IUCN, Walter and Gillett in 1998, 34,000 species, 12%, more than 12% species are threatened today. Now the figures are more higher only. The reduction in abundance of populations which is taking place. See the diversity. Anybody can get really engrossed in biodiversity or plant diversity of the planet. So these are the diversity. Loss of biodiversity is a major problem, and you can see these flowers. Do you want to lose them permanently? So we don't want to lose them, right? So taxonomy has many impediments to manage and use biodiversity. So global taxonomic initiative for capacity building has been taken up, and we have insufficient funds, resource towards plant conservation, and therefore we need funding also. So Systematic Agenda 2000 is a new initiative by USA biologists they have taken, and in 25 years, we have decided to discover, document, and inventory global species diversity to synthesize the resulting data into a predictive classification and to develop an appropriate information system to handle the resulting information. This we decided in 1994, but in 2020, we have not achieved the first goal to discover and document past and present life diversity. Okay, second, analyze and synthesize information. Then third is understand the evolutionary mechanisms. And fourth is to communicate and apply this knowledge to science and society. So these are the initiatives taken by the world, and this is the way cooperation from all: charting of biodiversity, taxonomists, ecologists, microbiologists, then developing some system of classification, understanding evolutionary processes, and then removing taxonomic impediments, funding. All this together with international cooperation, we can really think of making databases and retrieval of information, conservation and sustainable utilization of biodiversity. Okay, so biodiversity taxonomists, ecologists, all these are very important. Now in CBD, these are the articles. Okay, these are articles. I will not read because of time also. Time is a constraint. These are the articles. For example, develop national biodiversity strategies and action plan, Article 6, identification and monitoring, Article 7, in situ conservation, Article 8, ex situ conservation, Article 9. All these articles, sustainable use. Without taxonomic knowledge, we cannot implement CBD. So that is these are the articles. Many are there. Without our knowledge of taxonomy, we cannot implement them. So though it is a very important international treaty, without taxonomic information, we cannot implement. Now see this diversity of plants. Okay, we enjoy. And world has already started a wild seed bank and taxonomy. We are trying to conserve our biodiversity with that. Now we come to Western Ghats, where we are. We talk a lot of things about world, but what's about our own area, that is Western Ghats. This is the Western Ghats, which is a strip about 1900 kilometers long, and it plays an important role in hydrological cycle, climatology, and it has rich biodiversity. So it occupies only 5% of total area of the country, but possesses 26 to 27% of biodiversity. That means it is the strip with highest biodiversity. It is a treasure of biodiversity. And all of us, we have major developmental programs in Western Ghats: roads, windmills, sugarcane factories, agriculture. So most of cities we want to settle in this Western Ghat, which is a most important part for us. And it has a very high number of endemic plants, but nobody understands. So in Western Ghats, we have total 7,400 species, of which 5,500 or 5,600 are indigenous species, and more than 2,200 species are endemic to this region, and 1,273 are exclusively endemic to Western Ghats. So this is the data one should have. And this Millennium Seed Bank project conceived by Royal Botanical Garden, and they have now conserved plants. 34,088 wild plant species seeds have been conserved for future. So almost 13% flora is conserved in the form of seeds. This is the initiative. And another initiative which is taken is by Svalbard Global Seed Vault, where they have a capacity to conserve 4.5 million seed samples, and they are doing it every year. But in India, I don't find such a initiative. So anyway, these are the programs. Okay. Now I come to educational institute. We are in colleges, universities, schools. All my request to all the teachers and student is this one: educational and research institute have great role to play in conservation. What is the reason? The reason is your teachers train you and introduce biodiversity. They train you in identification and documentation of biodiversity. In biology, we train them, create awareness about biodiversity and its importance. Research scholars do scientific research and bioprospect our bioresources. And that way, major work on biodiversity is done by our educational institutes, and therefore we have great role to play. And why our students are very important. You know, this is a group of students searching for plants, rare plants on the plateaus. This is a Konkan plateau. They are searching for some plant which is a basal angiosperm, and that way we are training our students about biodiversity. Students, they have a strength, their enthusiasm, energy, and potential. They have love for nature. They have scientific knowledge of biodiversity. They have strong wish to care for Mother Earth. They have knowledge of plants, their identification, propagation, and multiplication. They have up-to-date knowledge of technological innovations which can be used for conservation of biodiversity. Innovative minds, enormous energy, enthusiasm, and strong desire to work. And therefore, I say, I have great hope and confidence that students, research scholars, and teaching community are playing and will play a crucial role in creation of awareness in society, documentation, bioprospecting, and conservation of biodiversity. And that's why this conference definitely will help our understand biodiversity. Hello? Kindly mute. Kindly mute.
Hello, Mr. Pungaviyar. Mr. Pungaviyar, please. Hello.
Hello.
Pungaviyar, please mute yourself.
Actually, Arun, you can mute all no from your side.
I mute. Is there any option? I don't know.
Arun sir, Chandore sir, you please mute everyone except sir.
Reclaim karo sir.
Arun sir.
Hello, Pungaviyar, Mr.
In some of the Indian Institute of Industry, all the PhDs are
reclaim करा document साठी परत.
all the PhDs very but now our
Hello. I have muted him. Right now, are you able to listen me?
Yes, sir. Yes, sir.
Yes, sir.
Okay. Now I could do it. Anyway, so what I was telling, our educational institute have great role to play in conservation, to create awareness and bioprospect our bioresources. And that's why, you know, schools, colleges, and universities have great strength of talented, hardworking, enthusiastic, and passionate students who can be trusted upon for conservation of plant diversity. This is what I feel as my teacher. Okay, so this is floristic diversity of our country. Of course, I will not go to all the figures, but angiosperms, total world 2.95 lakhs, it is said. And in India, we have now, this is the latest figure, 21,849 species of flowering plant. This is the estimate. And flowering plants are more important. Of course, other groups are equally important, but from angiosperms, we get maximum things, and that's why it is important. Educational institute and research institute, they do academic works. Okay, they create piece of knowledge. Okay, it may not be direct, it may not have direct applications, that is academics. Then we educate our students, our society with reference to biodiversity and many other things. Similarly, we do research. Similarly, we help how to utilize our bioresources without reducing them in nature, creating awareness and conservation. These are the functions of educational institutes. Now, these are the questions: what to conserve, why to conserve, when to conserve, where to conserve, how to conserve. All these questions have to be answered only by you and me, biologists. Okay. Now, I will give some example. It is not just a giving lecture, but some work which I have done. So our Dr. Malpure referred about Lead Botanical Garden, Shivaji University, all of you know, and in Shivaji University, 35 years before we started developing a botanical garden, university authorities gave full cooperation, provided land of about 42 acres land to the botany department, and every university has 300, 400 acres of land under their jurisdiction, and they can really allow this land for botanical garden. And we started developing botanical garden. The first aspect which we did is collecting seed banks. So per year we collect the seeds of about 250 species, and these seeds, we collect from nature. But in nature, all these seeds go waste. They are attacked by fungi, insects, birds, and no seedlings are established. But if we bring them to the laboratory and grow them, we can raise thousands of seedlings. Now you can see in Shivaji University, we raise these thousands of seedlings in nurseries, and each species is represented by many seedlings. Now these seedlings, we give it to our Gram Panchayat, municipal corporation, educational institute, colleges, universities, research institute. We distribute these seedlings. Don't you think that every college, every university can do it? Because we have knowledge of plants, where they grow, when they flower, when they seed, when to collect seeds, how to germinate seeds, how to raise saplings. All this knowledge is with biologists and educational institute. So our role is a primary role in conservation. In Shivaji University, we have raised, one palmatum in palms, more than 150, more than 110 species of palms are there in our Lead Botanical Garden, which are collected in last 30 years and planted in botanical garden. And we have thousands of seedlings of palms with us so that anybody can grow. Similarly, in Lead Botanical Garden, we developed an arboretum of endemic tree species of Western Ghats. 100 tree species, each with 9 individuals, have been planted in Lead Botanical Garden. And this is first kind of arboretum representing 100 endemic tree species of Western Ghats at one place. Now student can come, student can study, student can do bioprospecting, and that way it helps a lot in education and research. You can see these seedlings. I need not to say, with your own eyes, you can see the seedlings and this nursery. Now, this monotypic endemic tree genera like Irinocarpus pneumoni, Blepharistema, Ceratum, Utonepalium, Stipulaceum, Agasthymalaya, Pauciflora, Plesionaram, Indicum. These monotypic endemic tree genera, I know none of you who are listening me probably might have seen this genera. These are monotypic, endemic, restricted to Western Ghats. And if we bring them in the botanical gardens, all our students will come to know what are the endemic tree species of Western Ghats, what are the monotypic trees of Western Ghats, and what way we can utilize them for our benefits or for how we can conserve them. So this has been done by Shivaji University. See the palms, already I have told you, more than 110 species of palms we have brought in botanical garden, and that too by collecting seeds from all over the country and bringing seeds, germinating them and planting them. Every college can do it. Remember, every college can do it. Every university can do it. Only thing which you need will and passion with you. And you can also contribute to the conservation of biodiversity to a great extent. Now, another program which restoration of critically endangered species. Now you can see your Professor Arun Chandore. Here is Arun Chandore. Here is myself and other people, Guru sir. So recovery of 2 critically endangered plant species like Ceropedia fantastica and Hobardea heptanuran. With life risk in these crevices, we have snakes, scorpions, pit vipers, and our students have taken efforts to reintroduce this grass in various parts. And now you can see this, which was restricted to one place. Now it is growing at 100 places in 25 ghat areas, and more than 10,000 plants have been established. Don't you feel that it is a very important contribution and successful story of restoration of a critically endangered grass from India? So similarly, this is the Abitulan ranadi plant endemic to Maharashtra, northern western Ghat. Our student Anjali Nalavde, by artificial pollination, she created 45,000 seeds, raised saplings, and then with the help of forest department and students, they have gone back and planted in nature. Don't you think that it is a very important contribution? So these students, our student is our strength, and we can do wonders in conservation of biodiversity. We give seedlings to forest department, municipal corporations, and that's why colleges and universities are really temples where we can do many, many good things for welfare of Mother Earth. So you can see these people, right? Our Malis, our students, forest officials. This is Sharad Kamble, who is taking these plants to mountains and planting them, and such a wonderful experience I had with my students, and I feel really proud of them. Now you see, these are the Ceropedias. I will ask you a question, whether these Ceropedias are known by our Chief Minister, our Home Minister, our Prime Minister. Who knows this? Only biologists. You and me, we know these Ceropedias. Each Ceropedia is as important as a tiger. Each one is a genome, distinct genome. Who will conserve them? Again, your students are wonderful students. Technology, biotechnology, using biotechnological tools, they have raised plants through tissue culture, thousands of plants through tissue culture. Also, they have raised the plants through conventional methods, and then they have gone to the forest department and tried to restore plants in the nature. See this commitment of our students, and therefore I have great. So kindly mute. Hello. Hello.
Yes, sir. Okay, you can continue, sir.
See this transplantation program, which was taken by our student with their own money for conservation of 2 endangered species. So these are the things which they can do. And student told him that we have described many new species. Now it is 78. All these species are described by our students, which are new discoveries and documentation of novelties from our region. Okay. So now we see discovery of new species. It is not just a discovery of new species. We discovered a Mucuna sanjapti, which has L-DOPA 7%, while Mucuna prurens, source for L-DOPA, is having 3%. So it is a very important resource for L-DOPA. Okay. Now another example is our crinum. This is a crinum malabaricum, which we described. It has longest leaves in genus Crinum. Its leaves grow approximately 5 centimeters per day. Then it is restricted to 0.5 square kilometers in the entire world. This is a very important thing. We need immediate priority for conservation. Its allied species is Thyanum. It is a tetraploid, and it has highest content of galanthamine but it is a very rare plant. And this way, new discoveries have many other implications which one should try to know, right? So anyway, Trithuria conconensis, this is academic contribution. This species was described by myself and Dr. Janardhanam. This is Trithuria conconensis we named, and now it is considered to be a basal angiosperm. So any Indian flora written as per APG4, it will start with this plant, Trithuria conconensis, family Hadatilaceae. Okay, so you see this systematic position. So this is closely related to Lupar, Nymphia, Barclaya, but Hidatilaceae plant is only 1 centimeter total plant, but molecular biology has shown that it is closely allied to Nymphaceae. So this is a very important discovery from our country. Similarly, people, people always complain, our teachers, botany teachers, they don't try to know the plants around them, where they are working. And our Arun Chandore discovered 5 new species from a pond near Jaitapur, which is only about 50 square meter in size. So you can understand. Similarly, we can do wonderful work in bioprospecting of our plants. Delphinium mulbaricum, this was domesticated, partially domesticated and released as a floricultural crop for farmers. Okay. Similarly, Barlerias, you can see, which are growing in Lead Botanical Garden. So I gave you some few examples. I can keep on talking on this for a long time, but only some glimpses I have given that Lead Botanical Garden in Shivaji University is doing significant contributions in conservation and bioprospecting of our biodiversity. So plant diversity, I consider it is a biological capital of the world country, biological capital, which is probably most important or more important than any other capital, money, gold, any other capital. Biological capital is a very important capital of mankind. And, whenever we see at any species, remember, every species has many other aspects to be understood. It is one thing, it is unique creation of nature. It is unparalleled chemist. It is witness of evolutionary history. It is a phytogeographical significance, different functions in ecosystem. It has different range of tolerance, spatial relationships, and therefore one should really respect plants. Plants are, best friends, doctors, artists, designers, engineers, economists. Save the plants. Plants will save you. Okay. Plants will give you protection. And finally, plants are best teachers of our life. So in life, there are 4 things which are most important, 4 basic needs: water, food, medicine, and oxygen. All these 4, only water is different. The remaining 3 come from plants: food, medicine, and oxygen. So plants, I consider as the best scientist of the world. They only use minerals, water from soil, sunlight, and carbon dioxide from atmosphere. With this little capital, they give you food, they give you medicines, they give you oxygen, and therefore I consider them as a best scientist of the world. Okay, so if you conserve plants, we can really help Mother Nature. So in 1730, 373 people gave their neck to save the trees. They were knowing that trees are more important than their lives. And here the story, probably many of you must have heard before. Once again, I will repeat that God Krishna, Satyabhama wanted to weigh God Krishna with gold and make him, establish right on Krishna. So she tried to weigh Krishna with gold, but in spite of putting all her gold in this pan, Krishna was heavier than gold. Now, after her gold was over, Rukmini came and she took out all the gold and put one leaf of plant, that is, it is said Tulsi Patra, but it is an indicative of a plant. One leaf of plant, indicator of plant, which was placed in one pan, and this one leaf weighed heavier than God Krishna. So this leaf, one leaf, why it weighed heavier than God Krishna? Scientific is this one, that now I have told human survival depends on plants. We depend on plants for food, medicine, and oxygen, without which we cannot exist. And therefore, one leaf of plant weighed heavier than God Krishna. And our forefathers told us, oh man, if there is something more important than God for you, is a plant. And that is the scientific message from this story for us. Now, Mother Earth, how we are going to hand over our Mother Earth to our future generations is important aspect. We must keep all the resources of our Mother Earth intact and then hand over our Mother Earth to coming generations. And therefore, all of us, we are deeply concerned with conservation of biodiversity. And that is what I wanted to tell you in this lecture. So, challenge for 21st century is the conservation of biodiversity. Basic sciences are most important for understanding nature, and we are working for posterity, not for present generations. Okay. With these words, once again, I am thankful to the authorities of colleges, Dr. Bohr, principal of the college, Dr. Sandore, Sandan Shive, Professor Sandan Shive, for giving me opportunity to share my thinking about biodiversity, its conservation, bioprospecting, and sustainable use, and the role of taxonomy in this endeavor. Thank you very much for patient hearing. With these words, I think, thank you very much. Hello. Hello.
Thank you,
so much,
sir.
Okay, thank you.
Now I request to.
Thank you for your presentation. It was really great, sir. The response we're getting about your presentation is such a warming, and hope everyone is enjoyed and understand the value of biodiversity conservation and such a great presentation work. Your points such as the origin of human beings, reef mountains, biodiversity loss are really great. I would like to tell everyone that all the photographs were seen in the PPT was captured by Dr. S.R. Yadav sir itself. And also like to tell that the new genera from Western Ghat was introduced by Dr. Ian Sandore and others were named on the honor of Srirang Yadav sir as a Srirangiya. It was a really honor to heard you, sir. Thank you so much for such a valuable guidance.
Thank you, sir. Thank you.
Now I will request Dr. B.P. Gade sir for a vote of thanks.
Hello. Sir.
Yes, you are audible, sir.
Yeah, okay. Professor Yadav sir has given a very nice message. Save plants and plants will save you. Thank you for such a nice message, sir. On behalf of Rai Shikshan Sanstha's Art, Science and Commerce College, Mokhada, I would like to express my sincere thanks to Professor Yadav sir, eminent professor from Shivaji University, Kolhapur, for delivering his keynote address on role of plant systematics in documentation, conservation, management, and utilization of biodiversity. I hope this lecture will be very useful for our participants. Thank you once again, sir. I would also like to thank Dr. Malpure sir for chairing this session. Thank you, sir. Over to you, Taide sir. Taide sir.
Thank you, sir. Now I would request Dr. Shruti Kasana ma'am to introduce our today's invited talk, Dr. Asik Tamboli sir.
Thank you so much. Welcome you all again to the International Conference on Biodiversity, Conservation and Role of Molecular Phylogenetics in Plant Systematics. We had a great opportunity to listen to Yadav sir's lecture where he enlightened us towards our responsibility towards biodiversity and its conservation. Now we would be moving to the next session where I would like to introduce the invited speaker, that is Dr. Asif S. Tamboli, who is presently working as a full-time researcher at the Department of Biology, School of Life Sciences, Kongpuk National University, South Korea. Dr. Tamboli works in the field of advanced molecular systematics. He works in next generation sequencing, metagenomics, proteomics, including prediction of 3D protein models, to name a few. He is an alumni of Shivaji University, Kolhapur, from where he has done his graduation, post-graduation and PhD. For his doctoral degree, he worked on the molecular phylogeny and genetic diversity of important medicinal plants. He has also worked as a GRF and SRF in DBT funded project, followed by working as a CSIR SRF at Shivaji University, Kolhapur. He has worked extensively on plants, bacteria, aquatic macrophytes, etc., and has published his findings into national and international journals of repute. We are fortunate to have him with us as the invited speaker for the present session. I welcome Dr. Tamboli. Please deliver your lecture. Thank you.
Thank you, ma'am. Now over to Dr. Tamboli, sir. Dr. Tamboli, sir.
Hello, am I audible?
Yes, sir.
Yeah, is my screen is sharing?
Yes, sir, visible.
Yeah. First of all, thank you very much to the organizing committee of this conference to giving me opportunity to deliver the lecture on the today's topic, role of comparative genomics and phylogenetics in plant systematics. So before going to start to my today's talk, first of all, I am to tell you that I am from biochemistry and biotechnology background, and I am collaboratively working with the plant taxonomist and animal taxonomist to solve the problems in plant systematics as well as the animal systematics. So in today's talk, I am going to cover that what is the role of comparative genomics, what is the process of phylogenetics, how the DNA sequencing is carried out, and currently what scenario and what genomics techniques has been utilized to solve the systematics problems. So let's begin with the comparative genomics. So comparative genomics is the field of biology where different genome from different organism or different genes from different organism compared with each other to gain some evolutionary and molecular differences between the species. Now, what is the phylogenetic analysis? So it is the method of inferring or estimating the evolutionary relationship among the organism by comparing same gene from different organism, and it gives you a meaningful information about the evolution as well as process of evolution. And we know that angiosperm is the first plant group which is reclassified based on molecular phylogenetic data. So now what are the general steps involved in the phylogenetic analysis? So when you decide that you want to study the phylogeny of particular organism, what are the steps you have to follow? First is the selection of organism, whether it is plant, microorganism, insect, or any animal group. So after deciding your targeted organism, you have to choose appropriate molecular marker. Now, how should you choose the appropriate molecular marker? So in case of bacteria, 16S rRNA gene is a suitable marker. In case of animal, cytochrome oxidase gene is a suitable marker. But in case of plant, there are variety of molecular markers are available. So I will talk in detail on this latter, how to choose the appropriate molecular marker to study the plant phylogeny. So once you confirm your molecular marker, you have to amplify that gene with the help of PCR, and after the amplification of that gene, you proceed further for its sequencing. And once you perform the sequencing, last step is to analyze the sequencing data and its assembly. So these all steps comes under the genomics parts. From this point forward, the steps started with the molecular phylogenetic studies. So suppose you selected a plant group in which there are 10 species and you have amplified one gene from all the 10 species. You have to align all the 10 sequences, which is that called as multiple sequence alignments. So after that, you have to choose a suitable evolutionary model for your data, and then construction and evaluation of phylogenetic tree, and then interpretation of the tree. So these steps comes under the phylogenetic analysis. Now, as I told you that in case of plant, there are number of molecular markers available. So how to choose among them? Now, we all of know that plant possesses three kinds of genomes: nuclear, mitochondrial, and chloroplast. So first of all, you have to extract the total genomic DNA. Total genomic DNA means that total genomic DNA will contain all the nuclear, mitochondrial, and chloroplast genes. So nuclear genes are mostly favored for plant molecular phylogeny because the evolutionary rate in the nuclear genes are higher as compared to chloroplast and mitochondrial genes. And mitochondrial genome is highly conserved. So most of the studies or most of the researcher favors nuclear and chloroplast gene for plant molecular phylogenetic studies. In chloroplast genes, there are some coding and non-coding regions available to be used as a suitable molecular marker. In nuclear gene, internal transcribed spacer or external transcribed spacer, these two genes mostly favored. And in chloroplast gene, coding regions such as RBCL, MATK, and some non-coding region like tRNH, PSBA, tRNLF, and tRNSG. These are the intergenic spacer that can be used for molecular phylogenetic studies. Now, how to construct the molecular phylogeny? There are two types of methods: character-based methods and distance method methods used for phylogenetic construction. But nowadays, only character-based methods are favored for constructing the phylogeny. Now, when deciding the molecular marker, there are some criteria that your molecular marker should possess the following properties. And there are three properties that molecular marker should possess. One is universality. Suppose you selected one plant group, that gene should be present in the every species of that plant group, and it will be easy for its amplification and sequencing. Second is the sequence quality. When you perform the sequencing run, it resulted into the good quality of sequence. And third one is a discriminatory power. That gene should possess good amount or enough amount of variable sites that can be used to discriminate one species from another species. So these three property your marker should possess. Now, how to automated DNA sequencing and analysis? So suppose you have decided to amplify any gene from your plant group. So first step is the selection of plant material. So we generally use young and fresh leaves as a starting material to extract the DNA. So you can extract the DNA by using manual method or kit-based methods. After DNA extraction, you have to perform the PCR amplification of your targeted gene. Then you have to purify that PCR amplified product. After that, you have to apply Sanger sequencing principle to carry out the DNA sequencing of your desired gene. And after that, again, you have to perform one purification step to remove other impurities. And final step is the sequence detection. That means you have to load your samples on DNA sequencing machine, and then data will be generated. And last step is the analysis of that data. We will go all of the these steps in detail. So first one is the extraction of DNA. So you can see here the image of agarose gel electrophoresis. 1% agarose gel electrophoresis carried out to confirm whether your DNA extraction is successful or not. So I highlighted the DNA bands here. So you can extract the DNA by using manual method, or there are lots of commercial kits available to extract the DNA from plant leaf material. Next step is the PCR amplification. You have to load a 100 base pair ladder to confirm the size of your amplified gene. And here at the bottom of the image, you can see that there are some primer dimer formation, and these are the impurities you have to remove before going for the sequencing. In the right side image, you can see that it is after purification. You can see that there are impurities removed, primer dimer are removed after the purification. So in both the cases, you have to perform agar-agglutinative forces to confirm that whether your PCR product is purified or not purified. So for that, there are commercial kit available to purify your PCR product. It is generally silica-based column purification. Your amplified PCR product is attached to that silica column and other impurities are removed. And finally, you elute your PCR product by using buffer or nucleus-free water. And after that, you have to perform agar-agglutinative forces to confirm that all the impurities removed, to confirm that whether your impurities removed or not. So after performing the purification of PCR product, next step is the application of Sanger sequencing principle. So when Sanger discovered the principle, he used radio labeled DDNTPs, and he carried out polyacromate gelative forces, and after that autoradiography to detect the band. So what is the Sanger principle of sequencing? When instead of DNTPs, DDNTP is incorporated in the reaction, chain termination occurs and different fragments were generated. And these fragments is resolved on polyacromate gelative forces, and reading those fragments from bottom to top gives you 5' to 3' direction. In automated DNA sequencing, few steps are modified. The principle is same, but instead of radio labeled DNTPs, currently fluorescent labeled DDNTPs is used. Every DDNTP is marked with or attached with a different colored fluorescent dye. And what happens in the cycle sequencing reaction, instead of normal DNTPs, when fluorescent labeled DNTPs is used, chain termination occurs. This step is called as the cycle sequencing, where we applied the Sanger sequencing principle. And you have to again perform the purification to remove unutilized DNTPs, unutilized DDNTPs, and you have to load that sample on DNA sequencing machine. Now what happens after loading of your samples on DNA sequencing machine? Instead of pH, when Sanger discovered his method, he used polyacromate gelative forces. Here we use capillary electroforces. The resolution power of capillary electric forces is 1 base pair. So every fragment is resolved according to 1 base pair resolution, and terminal DDNTPs were detected by fluorescent detector, and your sequence will be recorded in the form of ATJC in the computer associated with that machine, and you can withdraw that data and analyze by using some bioinformatics tools. So the read length, you can carry out the sequencing of a gene having length up to 1000 base pair. You can carry out that sequencing by sequencing of that gene by using Sanger sequencing method or automated DNA sequencing method. Automated DNA sequencing is the it's a automated DNA sequencing machine implies or it used the same principle that Sanger have discovered, but few operations are automatically happens. That's why it is called as automated DNA sequencing. And you carried out all these operations through one software which is installed in the computer associated with that machine, and through that you can use all the operation on that machine. So this is the interior parts of that machine. This is the sequencing plate where you have to load your sample. These are the capillaries where the capillary electrophoresis is carried out. And here is the fluorescent detector which records the signals, fluorescent signals, and it transfers to the computer associated with it. And that raw data is generated after the sequencing run. And after the generation of raw data, you have to carry out some bioinformatic analysis on that raw data to finalize your sequence. There are lots of various tools are available to carry out such a sequencing analysis, but I have mentioned two softwares here which I generally use, Sequencer and Chromas Pro. These are two softwares which can be used for DNA sequencing analysis. And after analyzing your data, you get a final sequence and you can perform nucleotide blast search to confirm the identity of your organism or to find out the similarity. So then after generation of sequencing data, you analyze that data and finalize your sequence, but you have to submit that gene sequence to NCBI Gene Bank database, and you will receive some gene bank accession numbers, nucleotide accession numbers, and that you can use in your research papers. So how you can do the submission of your generated sequences? So Bankit is one of the sequence submission tool. So you have to first create account on that Bankit submission tool, and you have to click on which kind of category your sequences belongs, and then you have to start your submission that providing some details. Then you have to mention some authors which are involved in the carried out in the sequencing of that gene, then publication status, whether your article is published, unpublished, or in press, then how many authors are involved in your publications, etc. Details you have to provide. Then you have to provide which kind of sequencing technology you use to carry out the sequencing. So first one is Sanger dideoxy sequencing. There are some other method also listed there, and what are those methods I will explain it to later. Then you have to provide your all the sequences in FASTA file format, and then you have to provide whether the sequencing is carried out by yourself or you outsource that sample. Then you have to provide some details of your plant material, means its collection site, geographical location, voucher IDs, etc. Species, genus species name also you have to provide. Then finally you have to give some annotation. Annotation means you have to provide whether your genes are coding region or non-coding region. If it is coding region, then you have to perform its translation, means amino acid sequence also. And if it is non-coding, then you have to fill another form. Then you have to submit your sequences to GenBank database. So after that, GenBank staff will analyze your details and provide, if all is okay with your data and submission details, they provide you some nucleotide accession numbers, and those accession numbers you can use even in research articles or any further studies. Then how this DNA sequencing and Sanger sequencing based phylogeny is used in plant systematics fields. So there are lots of application. Some of them application I have mentioned here. I will explain them through sharing my research, which I up to date done in the field of plant systematics. So first one is the family or genus level phylogenetic studies or biogeographic studies, divergence time analysis studies. So first study I have carried out on genus Cliome. When I started to work on this genome, our team recognized that some of the species of Cliome is segregated into the another genre. Few were them segregated into Coriandra and Cliocerata. So our intention behind this study is to analyze the phylogenetic relationship of the species from these three genera. So we collected some species from all which belongs to all the three genera, and we targeted some plastid and nuclear region. From chloroplast we targeted RBCL and MATK, and from nuclear region we targeted ITS region. So we carried out amplification and sequencing of this region and submitted sequences to NCBI GenBank database. And these are the nucleotide accession numbers you can see here. And these are the area of collections, and these are the plant species. So after that, you can see here the PCR amplification images of RBCL, MATK, and ITS region. After that, we constructed the phylogeny based on each region, and we realized that some of the Cliome species and Coriandra species are not yet resolved by the DNA evidence. So another main conclusion we found that the Coriandra viscosa, which actually belongs to Coriandra genus, but not showing the close relationship with the Coriandra species. So we concluded that this family needs a major attention to resolve this issue. Then our Indian team and some Australian researchers come together to sort it out this issue, and we constructed a family level phylogeny by increasing some other genes. So initial my phylogeny was based on 3 regions. We constructed a new phylogeny by increasing some sampling with the Indian and Australian Cliomacy member, and we constructed the phylogeny based on 4 different regions. And then we realized that whatever the Coriandra and Cliome species have close relationship, we merge all the Cliome species into the genus Coriandra, and the Coriandra viscosa, which is not showing close relationship with the Coriandra species, is now merged into the Arivella genus. So these are the some family or genus level application we have done in this Cliome genus or Cliomacy family. Another kind of study I have done, same study I have done on the genus Caparis. My target was a genus Caparis, but for that I have constructed the full family level phylogeny. So I have constructed the phylogeny of Caparici based on plastid region. The intention behind that is to analyze the relationship of Indian Caparis, because phylogenetically Indian Caparis was not that much of studied. So we want to know that whether Indian Caparis is closely related to the old world taxa or new world taxa. Now, botany people are aware of that. What is the old world or new world? Old world refers for the regions Australia, Asia, Africa, and new world is referred for Americas. So those species which belongs to America, Africa, Australia, we want to study whether Indian Caparis is closely related to the species from old world region or new world region. And the second objective was to analyze biogeographic history, means what is the ancestral area of genus Caparis, and from which region to which region the Caparis species has been dispersed. So such kind of objective we designed for our studies. We collected some Indian representatives of Caparis, and we targeted RBCL and MATK region for this study. And we submitted sequences to NCBI Gene Bank database. Here you can see that area of collection, its geographical location, herbarium number, and accession number for RBCL and MATK. So our phylogeny resulted that and revealed that Indian Caparis shows the close relationship to the old world taxa. And before our study in 2008, the major study was carried out on this Caparis genus, and at that time the researcher mentioned that whatever the species which belongs to new world Caparis clade should be merged into the another genre, or that species requires some new generic name, because these species not showing a close relationship with the Caparis. Because if you see that in Caparis clade and new world Caparoid clade, there is African Caparoid. So researcher put some efforts to renaming these species. Some new generic name were assigned to these new world Caparis species, and our phylogeny agrees with all the revision which have been carried out in the Caparici. And in 1824, Condole given one sectional classification for Caparis genus. That sectional classification is also perfectly reflected by our phylogenetic analysis. Now, another objective is to study the biogeography of Caparis. So what is meant by biogeographic studies? Mean to analyze or to predict or hypothesize ancestral area of your targeted plant group, and to describe that how the species has been dispersed from which region to which region. And if you have fossil record from that plant group, and if you calibrate on that phylogeny, that means divergence time analysis, and you exactly tell that how million years ago that dispersal has been occurred. So such studies belongs to divergence time analysis. So our biogeographic analysis revealed that Indian Caparices have a origin in Africa. I mean, from Africa, species has been dispersed to the other world, and Africa is the ancestral region for Caparis. So before that, in 2061 study also hypothesized same conclusion, but at that time, Indian Caparices was not included in their study. So we include our Indian Caparis species in that, and again reframe the study, reconstruct the phylogeny and biogeography, and reach to some conclusions. Another tribal level studies I have carried out, we targeted trib Mary which belongs to family Convallaceae. The reason behind to target this trib is because in Convallaceae there are 12 different tribs, but among the 12 different tribs, trib Mary is taxonomically complicated. So we targeted that trib to resolve some issues by using a phylogenetic approach. So in India, there are 22 taxa which belongs to trib Mary, and among them, Dystemic rhynchorhiza and Oporculana tanzaniensis is endemic to the India, and its phylogenetic position is not known to the world before our study. So this was our main objective, to provide the phylogenetic position of these two endemic species by reconstructing the phylogeny. So for that study, we targeted 3 regions, TRNF, MATK, and ITS region. This figure 1 is of Dystemic rhynchorhiza, figure 2 is of Oporculana tanzaniensis. These are some details of species, that area of collection, and NCBI nucleotide accession numbers for the region which we have targeted. And we construct the phylogeny based on these 3 regions. So in 2070, one revised classification for trib Mary was proposed, and our intention is to decide whether Indian species is agrees or that belongs to that particular group or not. So the one major revision was done on the group Merrimia SL sensu lato. This group is segregated into the 4 different groups. Merrimia sensu stricto, some species retained into Merrimia, but that group was narrowed. Some species were segregated into the Dystemic, some in Camonia, and some in Decalobanthus. So here I will explain our outputs by showing some different parts of phylogeny, because the whole phylogeny, I think that not much clearly visible. So first output is we collected one specimen which we called as Merrimia gangetica. In India, before our study, that specimen is called as Merrimia gangetica, but phylogenetic analysis and some morphological studies reveal that this species is not different than the emarginata. So that species should be called as Merrimia emarginata. So we added one taxonomic note in our study that this specimen should be called as Merrimia emarginata from this point forward. All the Genostagia accession which we have collected from India belongs to Genostagia group. Oporculana tanzaniensis, which is endemic to India, and its generic placement was in argument. So our study shows that it belongs to genus Oporculana, and it is closely related to the Oporculana terpetum. And one interesting fact we observed that in 2070, the revision which occurred, they merged Merrimia rhynchorhiza, Merrimia rhynchorhiza into the Dystemic rhynchorhiza. But our study shows that Dystemic rhynchorhiza, Camonia vitifolia, and Dystemic quinatus is in close relationship. And in our study, this relationship is not resolved, means whether Dystemic is not merged into the Dystemic or it should be merged into the Camonia. There is no that much of strong support we get, and its generic placement is still in argument, as well as Camonia vitifolia, which not placed into the Camonia group and showing close relationship to the Dystemic group. So these 2 species, that Dystemic rhynchorhiza and Camonia vitifolia, the further study which we needed to resolve this complex, as well as we got some unresolved relationship in Merrimia. But this unresolved relationship may be due to that some of the Chinese species, Chinese Meremia species is yet to be added into the phylogeny. And after addition of that species, maybe we get a clear picture of that. But here is one example that even Sanger sequencing based phylogenies have some limitation in the resolution of some relationship in some plant group. So I will explain how can we resolve such a kind of problem if your Sanger based phylogeny is not resolving your question, then what genomics approaches you have to use to solve such problems. Another is describing new genera or new taxa. I did one collaborative work with the respected Chandore sir. We collaboratively described one new species from genus Pogostemon. Pogostemon jaitarpurensis was the species name. Based on morphological and molecular evidences, we have described this species. It is closely allied with the Pogostemon deccanensis and Pogostemon erectus. So we provide some morphological differences in between these three species, and we targeted 5 different regions to construct the phylogeny, and we construct the phylogeny of whole Pogostemon genus, and we shown that this Pogostemon jaitarpurensis is a sister to erectus and deccanensis, and it takes a different position, and morphologically it is different, and phylogenetic support also tells that it is a different from these two species. Another new species, I have a contribution in describing a new species from a genus Biolaria. Biolaria lavania was the species name. In that study, we provide some morphotaxonomy, cytogenetics evidence, as well as phylogenetic proof also. We targeted RBCL and Matke region for that. And this is the image of Biolaria lavania. We provide some morphological differences with its closely allied species, which is Biolaria longiflora. And our constructed phylogeny shows that the Biolaria longiflora and lavania has some close relation, and this new species belongs to subgenus Biolaria. This phylogenetic approach is also utilized when you are going to describe any new genus. Till date, I don't have contributed in such a kind of study, but I am giving some examples here. The previous researcher done such a kind of study. But in case of animal systematics, I done some collaborative work with the animal systematics peoples to describe Wardia as a new genus based on morphology and phylogenetic study. Then resurrection of species. Now, what is the resurrection of species application? So we carried out one study on Quirky Braviphillia. So this species was previously merged into the Quirky orchidus, but we found some evidences that that Quirky Braviphillia is different than Quirky orchidus. And we give some morphological, cytological, and molecular evidences to prove that Quirky Braviphillia deserve a species rank. So we done the resurrection of Quirky Braviphillia in this study. For that, we targeted whole Hypoxodidae family. So previous studies done some work on Hypoxodidae. So we generate some RBCL and tRNA sequences from our Indian Quirky species, added them into the data previously generated, and constructed the phylogeny. Here some morphological differences in between Quirky orchidus and Braviphillia. And our constructed phylogeny shows that Quirky orchidus and Quirky Braviphillia is closely related of each other, but Quirky Braviphillia is closer than other Quirky species which we have sampled. And we claim that yes, Quirky Braviphillia deserve a species rank, and it should not be merged into the orchidus, Quirky orchidus. So this kind of study we called as a resurrection of a species. Here are some cytogenetic evidences to prove our point. Now, resolving species complexes. Up to date, I didn't yet contributed such kind of studies. Yeah, I am currently working on resolving one of the species complex, but for understanding, I give some examples of previous researcher. Now, there are some species in which there are some morphotypes are available. Means morphological, they looks like different, but still they have same genus species name. So we call the species complex to them. And if you want to resolve such a kind of complexes, then you can take a phylogenetic approach to resolve the species complex studies. Population genetic studies. Now population genetics means what? Suppose there is a one species in which you are interested, but you want to analyze the genetic differences among the population. Means suppose the one species is found in Maharashtra region also and some South state region also, and you collected some species, that species or population from Maharashtra and some South region, and you want to analyze the genetic differences among the population, then that kind of studies belongs to population genetic studies. So in that studies also you can take the help of phylogenetic approach. Now, as I told that whenever your Sanger sequencing based phylogeny fails to resolve your questions, then you can take or you can change your approach, or you can apply different genomics techniques to solve that issue. So after Sanger sequencing, lots of next generation sequencing platforms currently developed, and they are highly used in plant systematic studies also. So the pipeline for next generation sequencing studies is like that. The difference between Sanger sequencing and next generation studies is that if you want to sequence full genome of any organism by using Sanger sequencing method, then it will take a number of years to complete that job. But by using next generation sequencing, you can do that job within few months. So what is the pipeline for that? So you have to extract the DNA, then you have to fragment that DNA into the number of pieces, and you have to ligate some adapter whose sequence is already known to you, and that ligation to the DNA fragment is called as preparing sequencing library. And this sequencing library, you have to carried out sequencing on any next generation sequencing platform. I will explain you the different next generation platforms available in the market right now. So after that, a large amount of sequencing data will be generated, and that data you have to analyze using some bioinformatic tools. And finally, you will get the your entire genome if you are targeting to sequence entire genome of any organism, or in case of plant, chloroplast genome sequencing, or in case of animal, mitochondrial genome sequencing, you can done by using next generation sequencing. So these are some platforms: Roche 454 sequencing, Illumina sequencing, ABI solid system. These are non-Sanger based methods. Sanger sequencing is the first generation sequencing. These are second generation sequencing. But problem with the second generation sequencing is its read length. Means you can carry it is also called as a massively parallel sequencing. Means in a single run you can generate large amount of DNA sequencing data. But there are some other methods also developed, which we call as third generation sequencing. The read length of these technique read length of these sequencing techniques is as compared to second generation sequencing is high. So currently all the second and third generation sequencing are highly used in plant systematics field. Now how these NGS based methods are used? As I told that in case of Trimerimia, we unable to solve the relationship with the help of Sanger based phylogeny. So currently whole chloroplast genome is sequenced from your targeted plant group, and plastome based phylogenetic studies carried out to solve the issues. Another approach is a RAD sequencing, restriction enzyme based DNA sequencing is carried out, and based on that phylogeny is constructed to resolve the relationship. Now the next generation sequencing, when first time coming to market, the cost is very high to perform such tasks. Now there are lots of improvement in the cost is happen. So all those area which needs DNA based evidences or which needs DNA sequencing data are attracted toward the NGS based platform. Now, if you see the progress of genomics and phylogenetics in resolving plant systematic problems, so it begins with the discovery of Sanger sequencing. In 1977, Sanger sequencing developed a chain termination method, and after that, in 1990, its use for phylogenetic study has been started. For first time, single locus or single gene-based phylogenies were used to solve the problem. But later on, in some plant group, the relationship among the organism or plant groups were not sufficiently resolved by single locus phylogeny. So multi-locus phylogeny or multi-gene phylogeny has been started. And that time, second generation platforms were in development. And after that, third generation sequencing platforms comes in the market. But initially, the cost was high for all these platforms. So nowadays, in last few years, the cost is to reduced, or it comes at affordable price for a researcher who is working in that field. So currently, Sanger-based phylogenies are also used, in NGS-based plastome phylogenies or rad sequencing-based phylogenies are used to solve the systematics problems. Now, if anyone who want to achieve research career in plant molecular systematics, or those who currently working in this plant and want to design your molecular work, what you have to do to achieve that goals? First one is you have the knowledge of plant taxonomy, and second, expertise in molecular biology technique like plant DNA extraction, PCR, DNA sequencing, etc. And some knowledge of bioinformatics, means how to analyze DNA sequencing data, ingest data, tools which are used in phylogenetic analysis, biogeographic analysis, divergence time analysis, and the tools which is used in population genetic analysis. Sometimes you need to have some background about the computer languages, because ingest data analysis was done on Linux kind of operating system is required to analyze that data. So this field, plant molecular systematics, means it runs by collaboration, means in that study, one taxonomist and one person from molecular biology and bioinformatics is involved, and togetherly this field runs. But if any botanist alone want to achieve all the things, so these are three requirements to achieving the good research objective in the field of plant molecular systematics. So thank you very much to giving me opportunity to deliver on talk on this topic. I hope this lecture will be helpful the beginners or the graduate, postgraduate students, or the research students currently working in this field to design some strategies on their interested plant groups. Thank you very much.
Thank you so much, sir. Thank you so much, sir. It was a really nice, informative lecture, as the taxonomy and the classical botany is important. The applied parts are also important to make awareness about the taxonomy and systematics. Your explanation about the Sanger's method was really nice, and new researchers are really motivated from this lecture. Now, I would like to request Mr. Dr. S.K. Pawar, madam, to give vote of thanks. Over to Dr. S.K. Pawar, madam.
Yes, sir. Good afternoon to all.
Good afternoon, ma'am. You are audible.
Yes. Honorable Principal Dr. L.D. Bhor, sir. Honorable Professor Dr. S.R. Yadav, sir. Speaker of the session, Dr. Asif Tamboli, sir. Chairperson of the session, Dr. Shruti ma'am, and all the participants. It gives me immense pleasure to express vote of thanks of this session. I am very much thankful to Dr. Asif Tamboli, sir, for accepting our invitation and delivering an informative lecture on a role of comparative genomics and phylogenetics in plant systematics. I am also thankful to chairperson of this session, Dr. Shruti ma'am, for chairing the session. Last but not the least, I extend my vote of thanks to all the participants because your presence put the stars in our conference. Thank you. Thank you very much.
Yeah, thank you. Thank you very much.
Thank you so much, ma'am. Now we will gather after the some short breaks at sharp 1:30 p.m. After that, we will discuss and present our papers.
All participants are requested. The paper presentation session will be start sharp 1:30. Therefore, all are requested. Please come. Please join with same link at 1:30. Thank you.
Dr. All participants, on the behalf of Arts, Science and Commerce College, Mokhada. We are going to directly starting the first session. So this session will have the 30 papers. Along with myself, another person, Nilesh Madhav, he's going to adjourning the working of this session. So contributorily, we'll ask the one by one participant, and next participant should be ready for the presentation. So keep in mind, each presentation will have the 5 plus 2 minutes. So the 5 minutes. Makrand Aitawade, please share the screen.
One minute. I will make a host to Dr. Makrand Aitawade. Please wait 1 minute.
Please. Okay.
Okay, Makrand.
Okay, Dr. Makrand, your screen is visible now. You can present.
Hello. Hello, I am audible?
Yes, yes. Your voice is too low. Please set your mic.
Okay. I think now I am audible well.
Yes, yes.
Yes. Okay. Good afternoon, everybody. I am here to present my research paper on rain-ballistic mechanism of seed dispersal in Begonia crenata. I will read abstract first. Begonia crenata is one of the narrow endemic species of Western Ghats, India. It is unique in having special mechanism of seed dispersal through rainwater. In this paper, adaptations, metamorphosis of floral parts and mechanism of rain-ballistic dispersal of seed in Begonia crenata is studied in the field and documented. The rain-ballistic mechanism of seed dispersal is reported first time among Indian begonias in the present research work. Genus Begonia is sixth largest genus of flowering plant with more than 2,074 species. The genus is classified into 70 sections consisting of herbs, shrubs, and lianas distributed through the tropical and subtropical region of the world. 64 species from India, along with 9 infraspecific taxa and 1 doubtful species under 6 sections. Uddin reported in India, the family Begoniaceae is represented by single genus Begonia with about 56 species, of which 18 are included in IUCN category. Currently, there are 75 accepted names recorded for Begonia in India. In Western Ghats, genus is represented by 16 species. Begonia crenata is one of the narrow endemic species of Western Ghats. It shows significant variation with reference to leaf size and color of flower and fruits. Fruit of Begonia crenata is unique in all the studied species in Western Ghats, which is having mechanism of rain-ballistic dispersal. This is the plant Begonia crenata. This is the illustration, and these are the photographs of Begonia crenata. This is the seed of Begonia crenata. This is the same photograph which shows a detailed morphology of external surface of seed. Now I will show you. These are the variation in leaf size, shape, and margin. See the leaf variation in a single species. Then there is variation in perianth number in male flower as well as number of perianth in female flower also. Here you can see 4 and 5 petals in male flower, while 5, 6, 7 petals in female perianth, female flower. Here also you can see the variation in perianth number, that is from 3 to up to 7. In similar population, I have found this variation. Now we will discuss seed dispersal in genus Begonia. In genus Begonia, seeds are commonly dispersed by anemoballistically. In this type, capsule dies by pore or slit and gradually release the seed with shaking of capsule by wind. Sometimes special adaptations to anemochory also found. Then there are some reports of zoochory as well as endozoochory are there. Then Leng and Bowman suspected myrmecochory in epiphytic African begonia species in the section Tetraphila, which was later confirmed by and described by Wilde in 2002. Then there is report of diplochory. Then there are some rattlebird dispersal also there. And animal dispersal of fruit in genus Begonia, the seeds are ornamented, which is common character of Begoniaceae, while many of older Endozoosaurus African Begonia lack the ornamentation. The Begonia crenata belongs to section Lochia. The section Lochia included 5 species, in which fruits are covered with glandular hairs and the largest wing become fleshy. There is no report of mechanism of seed dispersal from this section till date. This is the first report of rain ballistic mechanism of seed dispersal in the section Lochia, that is in Begonia crenata, endemic species from Western Ghats, India. Now we see mechanism of seed dispersal in Begonia crenata. Begonia typically possesses 2 locular fruit with very small ventral wing and 1 long dorsal wing. After pollination, perianth become fleshy, thick, and remain attached to the fruit. They are persistent. The ventral wing modified into disc-like or cup-like structure to receive raindrops for dispersal of seed, that is rain ballistic dispersal. Here you can see the persistent perianth which become fleshy and thick. Then the ovary, as ovary matures, the pedicel curves, bend down, and increase in length also. The fruit, so that the fruit develops like cup-like structure, and wings. These wings face upward, while the longer wing and taper point downwards, and whole structure act as a pendulum. In this video, you can see how these fruits or fertilized female flowers are looks like pendulum. Then these raindrops trapped into this cup-like structure of mature fruit, and these accumulate weight, and with the help of pendulum-like long wing capsule, not up down like this. Just a minute, I will show you one video. Like this, when raindrop received by these female flowers, the capsule wall get dehisced through a longitudinal slit or line of dehiscence, and seed get dispersed. Like this, in Begonia crenata, rain ballistic mechanism of seed dispersal happen in the nature. These are the some references I have referred for my presentation of paper. I am thankful to organizers to give me opportunity to present my research work in this international conference. Thank you, one and all. Thank you, sir.
Thank you, Dr. Makarand. This is very informative paper. Now paper open for discussion. If anybody having any question regarding this paper, they can directly ask. Anybody? Okay. Thank you, Makran sir, for your informative paper. Now, would like to call the next participant, that is Ahirao and Patil. They are going to present identity and understanding of some plants in perspective of Indian bioculture and medicine. Is there? Hello? Hello? Is there Ahira and Patil? Okay. So next participant.
Sir, 1 minute. Dr. Airav is here.
Acha, acha.
Now you are host, Airav sir. If you want to present, you can share your slide.
Hey, Rao, sir.
I think he has some problem. Okay.
There might have some problems. So next participant, Bala Sahib S. Kale, assessment of plant biodiversity, Munjaba Hills, a link between plain and northwestern Ghats.
Actually, he has sent videos. Okay.
Okay. So next participant.
Our organizing team sharing here videos of Dr. Bala Sahib Kale. Please wait for few minutes.
Okay, okay.
Is this video is visible?
Yes, sir. Video is visible.
Yes, video is visible.
Audio is not there.
I think audio is very, very blur. We can't hear properly. Excuse me, sir. Audio is not audible. So please, if you increase the voice, then we can't heard properly.
The volume is full, sir.
Might have the problem in video.
So better we skip this presentation.
I think so. We should skip this presentation
because it's
yes, because of we can't hear properly and we can't understand properly. So the next presentation, that is Award Anil Pathak Nidhi. They are going to present analysis of chemical phylogeny of SPLTC, fingerprints in family Myrtaceae. Is there award and Pathak?
They also had a video, sir.
Okay. Please share the video. I think in this presentation also having some problem, audio problem.
You can skip this, no?
Yes, I think so. We should skip this. Next presentation, Muzaffar Sheikh and Kumud Dubey. They are going to present a new distributional record of Ficus zehala in Madhya Pradesh. Here also having some audio problem.
Actually, we will share this video at the end of session on our group.
That will be the better. If we share the video after both sessions, so that will be the good. Shall I call the next participant? That is Nadar Ranjanatham and Ashwini Deshpande. They are going to present a study on effect of air pollution on plants.
Is not present.
Okay. Next.
Next participant is presentation number 7, Dr. Kumar Vinod Gosavi on Charymorphology of the genus Pinda, an elegant endemic genus to the northern Western Ghats, India. Sir, you can present.
Okay, wait 1 minute. I will host to Dr. Gosavi.
Okay, you can present, sir.
Yes, sir.
We go, sir.
Good afternoon, respected all. So I'm going to present Charymorphology of the genus Pinda, an elegant endemic genus to the northern Western Ghats, India. So the genus Pinda are the endemic genus to the northern Western Ghats and was having the monotypic nature with the species that is Pinda concahnensis. So the genus shares the affinities with the Heracleum, but very distinct among the Heracleum by remarkable characters. Like if we see the single umbellate of the Pinda, it seems to be a single flower because of the marginal florets of the umbellate that having the exterior petal that is anomalously growing. So it being as a outer petals, that is a major characters of this genus Pinda. And another interesting characteristic in their fruits, the fruits are the unwinged in their lateral ribs. And if we see the sclerenchyma forming discontinuous layer above the seed cavity, and the dorsal surface of the endosperm somewhat growed under the viti. So these are the major characters. Based on that, Mukherjee and Constance, it's segregated as a separate generic status. So recently, we added one new species in the genus Pinda, that is Pinda srirangi, and that published in 2020 in the Nordic Journal of Botany. So now this genus contains the two species, and both species having the very distinct in their morphological characteristic. So you can see their leaf, the Pinda srirangi having the ternate leaf, biternate leaf, and having the subulate at the end of the leaf tip, while Pinda concahnensis, which having the ternate to pinnately compound leaf and having the acute at their leaf tip. So both species very distinct in their umbellate nature, as well as the hairs which present on the their rays as well as on the bracteoles. So by that we can easily distinguish both genera. Both species. Even both species can easily distinguish by the having the petal, their shape, size, and having the hairs. Likewise, the family Apiaceae played an important role for the distinguishing in the carpological characteristic. So the carpology, that means the both fruits are the very distinct in both species, and they showed the separate entities in the generic genus Pinda. So when we compare the both species Pinda concahnensis as well as srirangi with genus Heracleum, because of it showed the affinities with the Heracleum. So we have taken one example, that is Heracleum aquilagifolium. So both species of the Pinda shares some common characters, but that is very distinct from the genus Heracleum by the umbellate, their umbel, then marginal floret, that is also abnormal, or you can say the zygomorphic corolla in the genus Pinda, while the regular in the genus Heracleum. Even the sepals also very different in a Pinda as compared to the Heracleum. So if we see the distribution of the Pinda, so the Pinda concahnensis that is distributed throughout northern Western Ghats, while Pinda srirangi so far reported only its type locality, that is Harishchandragarh. So because of this genus is adapted at the elevated areas of the northern Western Ghats, and both species adapted in different habitats. Pinda concahnensis, if we see it adapted into the plateaus, elevated plateaus, while the Pinda srirangi that is adapted in hanging steep cliffed rock at elevated areas of the northern Western Ghats. You can see by the both comparison photos, we can easily distinguish the habitat of the both species of the Pinda. So because of it having the different kinds of the morphology as well as different kinds of the habitat, it attracts to check out the charomorphology and understand their relation and their adaptation. So in present communication, we have discussed the chromosome counts and charomorphological characters of the both species of the Pinda. So mitosis study we studied by the conventional method. The root tips we obtained from the germinated seed. in a control condition. We use the pre-treatment for the paradichlorobenzene for the pre-treated in cool temperature in for the 4 hours. Then we squash the root tips in propionic orsin stain. So we used Olympus 20i microscope for the studying the chromosome counts as well as for the photoplates. So we studied the 10 well separated somatic chromosome counts. And karyotypic analysis, we followed the Levin et al. Methodology. So if we see, this is the chromosome counts as well as the chromosome plates of the Pinda concahnensis as well as Pinda srirangi. So if we see, so the karyogram that having the very distinct in both species, although both species shares the common chromosome counts, that is 2n is equal to 22. That is basic chromosome number X is equal to 11. But based on the karyogram, we can easily distinguish the both species because of, if we see the Pinda concahnensis, it has the less metacentric chromosome counts as chromosome number as compared to the Pinda srirangi. The Pinda srirangi having the more metacentric and medium chromosome numbers. And if we see the symmetrical index, gradient index and total chromosome length. So the symmetrical index in Pinda concahnensis is less as compared to the Pinda srirangi. Gradient index also having the less as compared to the Pinda srirangi, and total chromosome length, that is more as compared to the Pinda srirangi. So the karomorphologically, the Pinda concahnensis shows the more advanced character as compared to the Pinda srirangi. However, the Pinda genus, it showed some common characters, that is the chromosome counts, that is monobasic chromosome number in both species, that is X is equal to 11 or 2n is equal to 22. And Stebin classification, that is fallen under the 2A class. So that is thing. And therefore, the karomorphologically, Pinda concahnensis is highly advanced than the Pinda srirangi. That is we have concluded in the present work. So we used these selected references. This is my acknowledgement. We are thankful to SRB CRG project for the financial support. Also thankful to Principal SPTR sir and RYK Science College, Nashik for the laboratory facilities. Thank you.
Thank you, Gosavi sir, for very much informative presentation on genus Pinda. Next presentation is presentation number 8, presented by Dr. S.S. Kambale sir on trees of Trambakeshwar, Nashik district, Maharashtra.
Thank you, Nilesh. Is it possible to share my screen?
Just sir, wait, please 1 minute. I will share. I will make a host to you.
Okay.
Okay, sir. Now you can.
Whether screen is visible?
Yes, sir.
Yes, sir.
Okay. So thank you, organizers, for giving me an opportunity to share whatever I have documented in last 6 years from the Trambakeshwar, which is supposed to be a holy place, sacred place. And as far as 12 Jyotirlingas is concerned, Trambakeshwar is one of them. And river Godavari originates in Trambakeshwar. So with my colleague, Dr. Gosavi sir, we have started documenting trees of Trambakeshwar. Of course, Dr. Chandor sir is also part of this ever since he joined this Mokhada college. So this is what is my study area from Maharashtra. Nashik district's western part is Trambakeshwar Tehsil. And if you go to the Brahmagiri hill, on the top of the Brahmagiri, there is one temple where from river Godavari originates, and this is the Dugarwadi waterfall. So diversity wise, though Trambakeshwar Tehsil is the northernmost part of the northern Western Ghats and very much dominated by the dry deciduous forests, still tree diversity is quite interesting. Of course, endemism wise, it is not that rich as like southern Western Ghats. Still, we have documented around 130 species of trees in last 6 years. So documenting tree species was not the original intention of our studies. We have started with documenting entire flora of this Trambakeshwar Tehsil. As this Trambakeshwar Tehsil has habitat diversity wise, it is quite rich. So this is what is we have documented. At present, more than 800 species of flowering plants have been reported. Actually, this number we are expecting more than 1,200 species, as there was only 800 plus species was reported from Nashik district before by Lakshmi Narasimhan sir and other workers reported around 1,200 species from Nashik district itself. However, we assume that there must be more than 1,800 species in Nashik district, out of which Trambakeshwar must have more than 1,200 species. And we have so far reached till 1,000 species. This is what is the classical distribution of the species in polypetally, gamopetally, apetally, and monocots. Certainly, polypetally has more number of flowering plants in Trambakeshwar. The number is quite 281. That is quite a huge number. And as a result, what we have documented so far is more than 4 or 5 new species we have reported from Trambakeshwar. And the Anjaneri, one of the another sacred place, the entire floristic survey has been made and more than 400 species have been reported. So this is what is really actually the work we are doing here. And though there are 130 species of trees we have reported, only 6 of them are endemic. So endemism wise, we do not have maximum endemic species. But in herbaceous endemic, if you consider herbaceous species, there are more than 140 species of flowering plants. They are endemic, which occurs in Trambakeshwar, if you consider herbaceous as well as some shrubby species. So these are the top 5 families in Trambakeshwar. Leguminous is certainly the topmost family, of course, in the world as far as in as well as in Trambakeshwar, and Poaceae is the one. The other families like Asteraceae, Acanthis, and Malvaceae, they are with 51, 33, and 28 species respectively. When we were surveying, these are the top 5 families with 3 species. Combretaceae is actually not having the much, but Leguminosae is the topmost family with 26 species. They are there with the trees. And Combretaceae, Malvaceae. Moraceae is one with having more species of the trees after Leguminosae. And these are the top 5 genera, wherein Ficus stands at the top with 10 species. Ficus, we consider it as a keystone species, and Trambak is a dry deciduous forest area, and that's why there are several species of Ficus we get. Terminales, of course, Terminalia, Bohnia, and Albizia, there are 4 species each, and Diospyros is 2. You can see the trend is there are not much species genera there occurs in Trambakeshwar, being dry deciduous forest. So some of the pictures I would like to show you, that these are the 130 species. Of course, all the photographs I am not going to show you. And this is Dillenia pentagina, which is typically deciduous forest member, which flowers at present. And this is with smallest flowers, and fruits are also smallest. Another species of Dillenia, we get Dillenia indica also, which is planted majority. Mangifera indica, more than 10 cultivars as well as wild races we have found from Trambakeshwar Tehsil. This is Biodiversity fovillata, which was in the beginning when we were surveying. I was surprised to see Biodiversity fovillata. It is not a typical Biodiversity. That's why initially it was described as a Paleostigma fovillatum. Being the flowers and trees, they are actually dioecious. Male and female trees are separate. So you are quite surprised to see the leaves are like Biodiversity. You can see the size of the leaves. They are quite bigger in size. And flowers were, you can see, unisexual. Male plants were separate, female plants were separate. And we were surprised to see that when this is only female, flower, how it is producing fruits. Then suddenly we found a male plant also nearby. So there are several trees of these plants in Trambakeshwar, but the only thing is there are very few female trees. Male trees are quite more, and that's why getting fruit set is very much difficult. This is Heriolina quincunocularis, which is an endemic species. Of course, Biodiversity fovillata, now it is been placed in Phainera, genus Phainera. So Biodiversity fovillata is actually getting shifted from Paleostigma to Biodiversity, then from Biodiversity to Phainera, and that is an endemic species of our Western Ghats. Heriolina quincunocularis is endemic species to our India. It is not only restricted to Western Ghats, but you get in peninsular India also. Heterophragma quadrilocular, though it is a common occurrence, it is an endemic species of our country. So this is one. Garuga pinnata, people are making pickles out of these fruits. Garuga, you can see some Burseraceae member, and people are using the leaves of this particular plant as an antidote over the scorpion bites in remote places of the Trambakeshwar Tehsil. Sterculia guttata, they call it as a cooker in Marathi, and they are eating the seeds of this the species. They are edible. There are other species of Sterculia also occurring. Sterculia villosa is there. These are some of the references which are followed for the identification and confirmation of the species. And I was telling you how these mangoes, different land races of the mangoes, they are there been now in the Torangan Ghat. If you visit, you will find this diversity of the mangoes in this Ghat, size wise, flavor wise, and fiber wise. There is a lot of diversity of these mangoes. I thank organizers for the opportunity they have rendered. Thank you one and all. If there are some questions, I will be like to answer. Thank you.
If there is any question or doubts, you can ask directly to the sir. Okay. Thank you, sir, for this wonderful presentation on tree diversity of Trambakeshwar. Next presentation is presentation number 9. Bhushan Pawar, Vaibhav Shinde, and Swapnil Wagh on floristic diversity of family Fabaceae from Satmar ranges of Nashik district. Bhushan Pawar, Vaibhav Shinde, and Swapnil Wagh.
Sir, please host me. Bhushan Pawar.
Yes.
Okay, you can present, sir.
Screen visible, sir?
Yeah.
Okay, I will start. First of all, very good afternoon to all of you. Myself, Bhushan Pawar. Today's my presenting topic is floristic diversity of family Fabaceae from Satmar ranges of Nashik district, Maharashtra. Under the guidance of Professor Swapnil Wagh in PG Department of Botany, SNGBR, KKH Art, SMGL Commerce, and SPH Science College, Sandwar, Nashik. Then introduce to my topic. Biodiversity is the entire diversity and life on the earth. The greater exploration for the flourishing survey of flora and forest areas have greater importance. Most of the work has been carried out on plateaus from Satara, Kolhapur, Pune, but very scanty work has been carried out on plateaus from Nasik district. So we selected this study area for the present survey. Then objective to my research to study the flourishing diversity of family Fabaceae from Satmala ranges, to understand the flowering period, microhabitat, preference and distribution of each species. Then we have material and methodology is used for the research. Study area of my research. The coordinates of Satmala ranges are given in the slide, and the height of the plateaus of 1,472 MSL mean sea level. Then field visits in methodology. Frequent field visits was carried out for collection of specimens. Then herbarium preparation by standard method Jain and Rao. Then identification of specimens. All collected specimens were identified by using flora of Nashik district. Then by Lakshmi Narasimham and B.D. Sharma in 1991. And other flora of Maharashtra state, N.P. Singh, S.K. Karthikeyan, and P.V. Prasanna, volume 1st and 2nd in 2001. Herbarium consultation are authenticated by BSI Pune and some expert in the field. Then systematic treatment, the collected specimens are arranged in APJ4 system of classification. Then nomenclature and citation of description of taxa, use online database like PO means Plant of World Online and IPN International Plants Name Index for checking the scientific names and citation of the taxa. The given slide we have prepared the checklist of the plants we have collected. Then in this concluded that the botanical name plateaus, habitat and phenology and endemic or IUCN status, then execute a number of the plants. My flourishing diversity family Fabaceae consists of 3 subfamilies. First is the family Cisalpinoideae. Cisalpinoideae contain the some number of plants. Then next one is the subfamily of family Mimosaideae. And then next subfamily of Papilionoideae. The number of species are least concerned in this family, like as Taxonomy aspera, this concern in this family, and other one. These all are the checklist of the plants. Then abbreviations are there for the short forms are used in the checklist. Likewise, CP for the Chandrashuri plateaus, and likewise habitats are used in checklist. There are some. We have statistical analysis of the dominant species, means Alicecarpus. We have Alicecarpus. They have consist large number of species. Eight species are consist. Then Crotalaria consists 7 species. Indigofera consist 7, and Vigna 5 species are consist, and the last one is the Abrus are only one single species are consist. Then phenology of species are there. Most of the phenology means flowering and fruiting are there in June to August month of period. Then the core flowering and fruiting are the March in May. Next, habit-wise distribution. The most habits on this family, the herb are there. Then the core habits are in climbers are there. Next, habitat-wise distribution, plateau-wise distribution. The most of the species are occurred in the Chandrashuri plateau. In 40 species are found in Chandrashuri plateau, and least are the Vadabari plateau, are 2 species only are found in this family. Then conclusion, the total species were collected which belonging to genera, 31 genera. Out of 67 species, 11 species were endemic to Maharashtra as well as India. According to IUCN category of red list of threatened species, 24 were least concerned and 1 is threatened. Ethnomedicinally, 7 species were used to cure various ailments. Then major threat to these species are over-organized, so there is need to conserve this special habitat. Then these photoplates are have added for the some species. First one is the Cassia tora, second one is the Acacia jacquemonti, third one is the Glivicidia cpm, and fourth one is Cassia fistula. Then next are all are the Vigna species. This last one is the Vigna candelensis are threatened in our plateaus. Acknowledgement. We are much thankful to our guide, Mr. Swapnil Wagh, sir, for their valuable guidance and support. We are also thankful to our college principal, Dr. G. Jain, sir, and Dr. Manoj P. Patel, sir, head of department of our college, for allowing us field visits and providing necessary facilities. Then I have some references cited by the Floras or other. Then thank you. If any kind of question, please ask me.
Presentation is open for discussion. If any questions or doubts are there, you can ask. Okay. Thank you, Pawar sir, for the nice presentation on floristic diversity of family Fabaceae from Satmar ranges. The next
Thanks,
presentation.
sir, for giving me chance of presenting of my research work.
Thank you. Next presentation is presentation number 10, de novo transcriptomy of Travanaria quinifolia by Talib Ali Momi.
Hello. Sir, my screen is not shareable.
Dr. Pawar sir, you can turn off your share screen. Pawar sir.
Talib Ali sir, you can share the screen now. You are the host now.
Okay, sir. Is my screen visible, sir?
Yes, sir.
Okay. Very good afternoon to one and all. The topic of my presentation is transcriptomic analysis of Taverniera quinifolia. I am working as a research scholar in Department of Botany, Faculty of Science, the MS University of Baroda, and my guide is Dr. Padmanabhi Shankar Nagar. So this is the whole image of Taverniera quinifolia. It is shown in the flowering stage and it's in a natural habitat. So why I have taken this plant? As Shastras have described two types of licorice. One is Thalaj, that is the Glycerasa glabra, and one is the Jalaj, that is Taverniera quinifolia. Yet the Jalaja plant, that is the aquatic plant, has not been yet described, or the only preliminary work has been done, and it is shown by Zor and Mangnota. So germplasm was collected of Taverniera quinifolia from Gujarat area, Gujarat state, the Dinodar forest from Kutch, and one Manjuba village from Rajkot, and Bagodra near Ahmedabad. So preliminary work has been done on the three germplasm, and we have found out that maximum content of glycerasm that is present in this plant has been identified from the germplasm of Bagodra, that is 0.153 milligram per gram, and the maximum biomass of the root was obtained. And for further analysis, we have taken the germplasm of Bagodra for our molecular analysis. This is the brief introduction in which Taverniera has generally 16 species, and it has called as an alternative to the Glycerasa glabra plant. And we all know that Glycerasa glabra has been used in many industries like food, herbal, cosmetic, nutraceuticals, and many more. So Taverniera quinifolia has opined a new alternative to the Glycerasa glabra in India. Only 16 proteins have been reported in the NCBI till now, and for this only our study has aimed to carry out the transcriptomic analysis to identify the functional genes in this plant. And preliminary analysis have shown that glycyrrhizin, liquatine, glabridine, and liquatiginin are present in the Glycyrrhizia glabra. And in Tavernera cunifolia, we have found that glycyrrhizin and along with that 27 phytocomponents have been isolated by one of our seniors, and her work has been given in 2015. These are the approx 27 phytocomponents that have been isolated from Tavernera cunifolia. So what is the main significance of comparing this plant Glycyrrhizia glabra with Tavernera cunifolia? The harvesting time for G. glabra is 3 years, and for Tavernera cunifolia it's 1 year. Soil type we are using acidic saline. The root production is 3 tons per acre, and here it is 4 to 5 tons. And the glycyrrhizin content is approx 15.88, and in Tavernera it is 13.20. It is less than Glycyrrhizia glabra, but we can use this plant as an alternative for this is the proposed pathway biosynthesis of glycerasm, which was given by Saki et al. In 2011. And in our plant, we have identified the 2,3-oxidosqualene and beta-amyrin. Along with that, 11 oxo-beta-amyrin and 30 hydroxy-beta-amyrin also we have found in our plant. And ultimately, this all oxidation process ultimately leads to the production of glycyrrhizin. So this is the preliminary work that has been done. We have estimated and validated the glycyrrhizin by LCMS and using the solvent system at butanol, acetic acid, and water. And we have used the standard glycyrrhizin to track in our TLC. And the molecular work, we have done the NGS analysis of this plant, and 2 runs were of Tavernera root were run in replicates, and we obtained 5.5 GB of data. After filtering the raw reads and everything, we have gathered 35,590 transcript. Total, the length of the sequence was around 1,49,028, 1 crore 49 lakh 28,000 approximately, and the maximum length of sequence was 5,670 with N50 was 441. And then we used different bioinformatics tools to analyze our raw sequence. These were the tools that were used. And ultimately mapping and annotation was done with the help of BLAST2Co. These are the project statistics. Our analysis was carried out. The total sequence or the transcript we got were around 35,590. With BLAST hit, it shows 27,884. With mapping, around 22,510 sequences were mapped, and with annotation, it was around 21,066 sequences. These are the tag distribution, which shows 27,884. Among them, the gene ontology data was database was used, in which 21,066 were assigned to the GeoSlim, and there were 7,000 sequences whose BLAST was not there, or no match was found, and 22,510 sequences were mapped from this. The best species distribution for my plant was heated with Medicagio trunculata, that is 18,734 blast hits were there from this plant, and the second was Cicer areatinum. All these plants matches were for the Fabaceae family. The top hit species distribution was Cicer areatinum and Medicagio trunculata. The mapping data source was Interprot, then Uniprot, Geocentral, GeoC, and Ensemble plant. These were the database for where gene ontologies were mapped from my data. So evidence code was given for this. These are the geo distribution for biological process, molecular function, and cellular components. Biosynthetic process was among the tops that were matched. And in molecular function, nucleotide binding was the first that was matched for our Tavernera plant. And here membrane, nucleus, and cytoplasm were used for the cellular components. So all the data, all the 35,590 unigenes that I have got, I have matched my data with Kyoto Encyclopedia of Gene and Genome, that is CAG, and we found that 496 KOIDs were generated from this whole transcriptome database. And from that, the main pathways genes that are the cis-terpenoid and triterpenoid biosynthesis, we found that 3 genes were matched from my plant, and there were 3 other genes that were obtained by the terpenoid backbone biosynthesis, and there were 1 gene which we have found from steroid biosynthesis, and 3 genes were obtained from the carotenoid biosynthesis. 1 was from the flavonoid biosynthesis. 2 genes were obtained from the monoterpenoid biosynthesis, and again 2 genes were obtained from the zeatin biosynthesis. This is the enzyme code distribution of our plant. From all the 35,590 transcripts, we have got the maximum enzyme match with our hydrolysis, followed by transferases and oxidoreductases. These are the top 15 transcription factor families that have been detected from my data. The highest match was WD40 like transcription factor. Thank you.
Thank you, sir. The presentation is open for discussion. If any doubts or questions are there, you can ask. Okay. Thank you, Momin sir, for sharing your informative research work with us on Travernaria cunifolia. The next presentation number is presentation number 11 by Agnes Kharath, Inventory of Butterfly Host Plants Diversity from Nashik. Dr. Agnes Kharath.
I think Dr. Kharath is not there. Shall we proceed the next presentation?
The next presentation number is 12, Sandra Balakrishnan, on analysis and evolution of bioenzymes derived from fruit peels of lemon, orange, and pineapple. Dr. Sandra Balakrishnan.
Sir, actually, Sandra Balakrishnan has sent a video to us. So we will play it later.
Okay. The next presentation number is 13 by Meenakshi Mahajan, Plant Diversity of Tree Species of Ferguson College Campus, Pune. Dr. Meenakshi Mahajan.
Meenakshi Mahajan also sent a video to us.
Okay. The next presentation number is 14 by Bendekar GD, Effect of Eutrophication on Bird Diversity of an Urban Lake, a case study of Pashan Lake at Pune.
Hello.
Bendekar sir, you are a host now. You can share your screen.
Okay, sir. Hello. Hello.
Hello. You are audible, sir.
Good afternoon, sir. Good afternoon, sir.
Good afternoon. You are audible, sir. You can start now.
My name is Govind Bendekar. I'm going to present oral presentation of my topic is effect of eutrophication on bird diversity of an urban lake, a case study of Pashan Lake at Pune. Introduction. The vast diversity of wildlife bird found in the wetlands, including birds, mammals, frogs, fishes, and insects and plants. And the wetlands are the ecological indicators of ecosystem. Most of the birds are ecological indicators of the ecosystem health. Pashan Lake is a old lake in Pune city, an excellent overwintering site for the bird watching. And recently, there is an artificial restoration event after eutrophication. Currently, the lake passes through yearly repeated introspection cycles and receives polluted water from the river Bangladesh. Long-term biodiversity monitoring studies of ecosystem important for successful conservation. And this study signifies the importance of conjunctive biodiversity studies for the wetland conservation. Materials and method. The study was carried out at Pasan Lake, Pune, and this is the Google Map image of the study site. Birds were data collection. The birds were sampled using the distance sampling line transaction method, and at the early in the morning between 6 a.m. to 10 a.m. Because it is the activity time of bird. Data were collected between June 2021 and April 2022, and this is dividing year into the monsoon from June to September. Post monsoon period is the October to November, and winter season from December to February, and summer is from March to May. Hello. 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 Hello.
Yes, sir.
Different equipments such as Nikon D600 camera and Nikon binocular 10x50x zoom was used for the to take photographs and observation purposes. The identification of bird was done by audiovisual cues, and species were identified by using field guide of Salim Ali, Grimet and Inskis. And for bird taxonomy and nomenclature, we followed the Provin et al. 2016. Results and discussion. The lake process through eutrophication in the winter season, and in the summer it was seen completely covered with the hyacinth vegetation. According to McDonald et al. 2006, eutrophication sometimes eutrophication is beneficial for the bird to a certain point, but after this there is a change in occur in the habitat on which they rely for food and inserting nesting sites. In our study, we found that the number of water birds are decreased from January 2022 to April 2022 as a hyacinth vegetation increased. The reason for the decline biodiversity, including the most of the birds, water birds, feeds upon aquatic organisms, but due to the organic pollutants, the eutrophication of lake take place, which results in the decrease in oxygen level of lake. Dissolved oxygen level of lake is decreased, which further affects the aquatic life such as fish, frogs, and invertebrates. The eutrophication of Pasan Lake lead to a decrease in the richness of aquatic birds. This is the images of discharge of sewage from city to the Pasan Lake, and this image is the discharge of pollutant, carry bags, etc. And this is the anthropogenic activity responsible for the eutrophication of the lake and decreased water diversity. This is the stages of eutrophication. Gradually, the first image A from June 2021, and the B, second is from November 2021, and the third image is from January 2022, and the D image from April 2022. This is the list of the table contains the list of the water birds whose number declined from June 2021 to April 2022 due to the eutrophication. These are the birds, water birds. The conclusion, we concluded that increased anthropogenic activity within the surrounding of Pasan Lake led to eutrophication of the lake, which declined in the bird diversity. Once the eutrophication occurs in any wetland, there is very difficult to maintain it in an original state. We suggest that regular monitoring of the lake can prevent further loss of biodiversity and restoration of the lake. These are some pictures I have shared from the Pasan Lake. These are the references of studies. We thank the Forest Office Pune for the permit to carry out field work at the Pasan Lake, and Department of Zoology, Scientific University, Pune, for providing research facilities. We thank
Hello. Okay. Now paper open for discussion. If anybody having any question regarding the topic, they can directly ask. Thank you, sir, for your nice presentation. Because of your hard work directly reflected on your presentation. Once more, thank you. Now the next presentation, Nilesh Madhav on genera of tribe Midae in India. Nilesh Madhav.
Yes, sir, I am present here. Can I share my screen, sir? Hello?
Please wait for 2 minutes. Here, electricity problem.
Okay, sir. Okay.
That's why our system is switch off.
Bendekar sir, please mute your voice.
बंद
Arun sir.